Thanks a lot for the very nice introductions, and it's a privilege to be here with you and share part of the research that I've been doing since I arrived at Stanford University. Today, I want to talk about ecological levers for health. That is actionable environmental and ecological drivers of infectious diseases of poverty that can lead to win-win outcomes for nature and people. And I wanted to start to do that by bringing you, at least visually, to some of the field sites where I work in lower basin of the Senegal rivers at the border with Mauritania. The reason why I wanted to do that, because now that we are, basically out of the COVID-19 emergency, the big pandemic, thanks to the development of the vaccines, I just want to bring your attention that there is a silent epidemic that is occurring day after day, year after year, for decades, and affect a large fraction of humanity, especially in tropical and subtropical countries. This is a pandemic that is caused by diseases that have an important environmental component in their transmission cycles. And when I talk about that, I'm sure that the first thing that comes to your mind is mosquitoes-borne disease and malaria and Zika. And I also want to mention that in addition to malaria and Zika and other diseases, there is a lot of other diseases. Some of them are part of the so-called neglected tropical diseases that are also very relevant. They're still vector-borne diseases. Sometimes they're still mosquitoes-borne diseases or diseases that are transmitted by flies as a vector, or snail-borne diseases like schistosomiasis, which is my disease of reference, or fasciolasis, or soil-transmitted helminthiasis, like hookworms and whipworms, and a range of mycobacteria that are ubiquitous in the environment, and occasionally they find the opportunity to jump and infect the human host. Now, this is a very complex slide, and the point that I want you to take here is the diversity of pathogen and parasites and of different non-human host and life history strategies that these parasites have. In spite of this diversity, they do share some common feature. They all have a complex transmission cycle with an environmental reservoir, okay? And sometimes these pathogen and parasites spill over to the human host. And in some cases, like in schistosomiasis, they are part of an obligate transmission cycle. So the parasites goes from infected people down to specific type of freshwater snails and back to people. And that's the way the cycle is closed. A lot of these free-living stages of non-human hosts and parasites are affected, of course, by climatic and environmental conditions. And so now that we're living in a world of global changes, this has huge effect on the patterns of distribution, emergence, and persistence of these diseases. In addition to that, these pathogen and parasites and their non-human hosts are not there in a vacuum, like in a biosafe level 2 lab. They are merged in a food webs of interactions. They are food for some other species. They take advantage of some other species. And it's very important to understand this web of interaction to basically predict the dynamics of this pathogen. Another thing that is important is the fact they are neglected tropical disease. That does not mean they are not important. Actually, they are 80% of the 560 WHO tracked pathogens. They have some environmental component in their transmission cycle. 40% of the world burden of infectious diseases. They affect more than 1.5 billion people, with 3 billion people at risk. They're mostly focused here in tropical and subtropical countries, but thanks to climate change and globalization, they're starting to trickle up in the high latitude. And so cutaneous leishmaniasis is now endemic in Texas, and last summer we have seen some stunted chain of transmission of malaria in Florida. So they are coming to us as well. There are diseases of poverty, because poverty is both a cause and the consequence of disease. And they basically engage in these persistent disease-driven poverty trap there. In addition to that, these multicellular parasites or these unicellular eukaryote parasites are also escape artists of the immune system. The schistosoma parasites that is represented here is a champion of that. They use proteins of their human host to camouflage and hide from the immune systems and stay in our body potentially for years. That means that for most of these parasites, these take a really long time to mount an immune response, if ever. And it's not only that, it's also very difficult to find vaccine. COVID-19 has been a revolution in the speed of which we can do that, and we are not nearly there for the majority of these parasites there. And even there are some vaccines now, they only have a relative coverage, they wine out, they need to be repeated. There's a lot of challenges in their application and delivery. Medical treatment usually remains the only possible things to do with anti-emitting drugs, and they do work well. They remove the parasite, but they are necessary but insufficient type of treatment because they do not protect from further infection of the virus. It is therefore crucial, and this is the point, to target also the environmental reservoir of the disease. This is a really wicked problem, and by only pursuing medical intervention, we have not gone really far in recent years with few exceptions. And looking at the environmental reservoir of the disease means also to understand the landscape in which humans and pathogens are embedded and how they interact with the different types, the water, the vegetation, and how they are affected by the species and the animals that are present in their environment. And this, once we understand this, we can deliver better treatment to try to basically interrupt both arms of transmission from people to the environment and from the environment back to people. So for an area discipline that is known as global health or public health, I believe that disease ecology can actually bring a huge contribution in understanding the dynamics of the disease and in delivering better intervention for both environmental monitoring and surveillance, and for instance also for biological control. And I want to briefly go through the two of them to show what we have been doing in these 10 years at Stanford. And to do that, I'll bring you back to my research site at the border with Mauritania in the lower basins of the Senegal River, where we've been working now for nearly 10 years, over 16 study villages and several field missions. We've been tracking 1,500 school-age children, and we've been doing a lot of work in that complex life cycle, studying schistosomiasis, and so how the parasites goes from people to the freshwater snails and back to people. And this has entailed countless hours of labor-intensive, energy-consuming work in the field, the old-fashioned field malacology that is crucial to understand the distribution of the species, and hours dissecting snails and identifying parasites in the lab. We found a clear association with certain type of vegetations that is widespread in this area. And the question that we had is how far is going to be the snail that is going to infect us? So in order to answer that question, we went first to Manu Prakash, who was independently studying the unique swimming ability of schistosoma parasite that is able to crawl in this very viscous water there. And in the desperate effort to stay afloat close to the surface. So we realized that this parasite is unable to cover very large distances, but can basically take a free ride by staying on the surface when the wind blows them around. So in order to test this hypothesis, we went to Oliver Fringers, and this is something that only within the School of Sustainability we could do that, to try to study the fluidodynamics of infective larvae into our sites. And along with the candidate graduate students, Brooke Poken, we have been basically studying this, idealized site geometry, flipped that around, and started to basically run this simulation for a wide variety of geometry and morphology of the sites and with assumptions, and basically prove that indeed snails that are also hundreds of meters away still have the possibility of contributing to infection rate. We also look at our site from a bird's eye view to try to map the morphology and the amount of vegetation, and we found that there was a lot more vegetation that we could see from the shore, and we can identify that, and we can classify and associate with the suitability and the probability of finding the parasite there. And so we have basically developed these ideas of precision mapping, which is similar to the idea of precision medicine, where we can target intervention by understanding basically the type of habitat at each transmission site. We now know what we can do at the very local level, but then when we zoom out at the regional level, we can't use drones any longer because they're not effective, and so we have to use different type of techniques. So we have been looking at high definition satellite imagery with World War II, for instance, and we have identified, thanks to a collaboration with the Human Centered Artificial Intelligence Center, on using deep learning to do segmentation, identify suitable habitat or much larger scales. And now, basically, we're putting together this pipeline of multi-scale integration for the behavior at the microscale, at the increasing scale, to understand what is the distribution at the regional and country level, and develop a network epidemiological model that allow to identify what to do where and in the most effective way. Talking about the type of intervention, I also want to mention that environmental control of schistosomiasis is not something new. It's been there for quite a while, either with engineered intervention, which are always fantastic, but takes time, they're capital intensive, or using chemicals to kill the snails, which has a lot of other environmental problems. So we look at the control of schistosomiasis by using biological agent, and we did this work with the Macrobrachium volenovi, a native species of Western Africa, a voracious predator of the snail, and through the equivalent of clinical control trials in the environment, we've been shown, and I won't show you any p-value, but that indeed prawns is able to control transmission. Because prawns are also part of the staple food and the diet in Western Africa, we also have helped to develop a commercial level hatchery in Saint Louis in Senegal that is basically now providing labor to and work opportunity and a training opportunity for students in the Master of Aquaculture there. And we're now exploring a set of other species that could be integrated, for instance, with rice agriculture in Senegal. Another alternative is why not just remove the vegetation rather than trying to target with natural enemies, with predators. And these ideas has been investigated along with a colleague from the University of Notre Dame, Jason Rohr, and we have been basically established this experiment in which large chunk of floating vegetation that is suitable habitat for the snails were removed from the water access point. And we show that indeed this has led to a reduction in transmission. Not too much surprisingly to some extent, but very effectively. But the next question here, which I think is even more interesting, and what do we do with all that vegetation? Okay. And so one thing that we look at that is that we can use that vegetation, the floating vegetation, to create compost for agriculture. And this provide basically crops, at the same level of using industrial fertilizers at only 1/10 of the price. Not only that, this can also be used for feed for livestock. And this, again, in this experiment, and show that can basically feed livestock at the cost that is only, a small fraction of the other type of feed. And last but not least, there are promising results also in using floating vegetation in biodigester for the production of biogas. And all these results has been shown very in details in these papers that we have recently published. So basically, all these ideas are part of what we call the 4 pillars of poverty alleviating ecological solutions that I represented here. So type of intervention that really integrate through disciplines, and rather than working in silos, try to do basically intervention that improve public health, which is the Sustainable Development Goals number 3. They can also good to increase food security, the Sustainable Development Goals number 2, to fight poverty by by fostering small-scale business and integrated agriculture and aquaculture, the number one of the Sustainable Development Goals, and in some cases even to restore ecosystem integrity, which is the number 14. So in this multiple win type of solution. And the problem here is basically to understand what other diseases can be approached with the same way. This is my last slide. I just want to say that the way ahead is really exciting. And I, along with my colleague Erin Mordecai, are launching this new initiative, the Healthy Planet, Healthy People, or also called the Disease Ecology on a Changing Planet, where we intend to leverage on our work and understanding of disease ecology to expand the work to many more diseases and engagement with stakeholders in the country where this is needed the most. And I'm looking forward to have some questions for you guys. Thanks a lot.